Welcome back. So just to review what we were doing in the last video, we were talking about arginine biosynthesis, which of course involves the urea cycle. And so we spent some videos talking about the reactions in the urea cycle, some of the mechanisms of the enzymes in there. What we're going to do in this video is we're going to look at sort of an application of arginine metabolism, and that's to look at the biosynthesis of a molecule referred to as creatine. Okay? And creatine has all sorts of implications in brain cells and in muscle cells, and mostly what it's for is it's for storing the high energy gamma phosphate of ATP in a form that doesn't allosterically inhibit a lot of the enzymes that are important for other aspects of metabolism. Okay? And this synthesis of creatine and creatine phosphate are going to be split into two different videos. So in this video, we're going to look at the first two enzymes in the pathway, which are arginine glycine amidinotransferase, also referred to as guanidinoacetate synthase. And then we're going to look at the next enzyme, which is guanidinoacetate and methyltransferase. So we're going to talk about, first of all, the mechanism, and then we're also going to talk about the regulation of the enzymes and some important implications in all of this. Okay, but the ultimate goal is to look at creatine synthesis, and in the next video, we'll look at the functions of creatine and creatine phosphate. Okay, now what I want to point your attention to is the fact that the beginning of creatine synthesis starts with this amino acid, which is called L-arginine. Okay, and L-arginine possesses a very important functional group. Let me circle it in purple. Okay, so the functional group I'm concerned with is basically this right here. So the functional group I'm concerned with is basically this guy right here. Okay. And of course, there are several resonance contributors that contribute overall, but this functional group and all its resonance contributors is called a guanidino group. This is a guanidino group, okay? And effectively what this first enzyme is going to do, guanidinoacetate synthase, is it's going to take a molecule of glycine and it's going to transfer at least this component of the guanidino group onto glycine. Okay, so this component right here, this has a special name. This is called an amidino group. Okay, it's basically part of the guanidino group, except one of the nitrogens is of course excluded from it. And so the reason you call this enzyme glycine amidinotransferase is because it's going to take this amidino group and transfer it onto the alpha amine of glycine. Okay, and that's actually what we're going to look at first is the mechanism of guanidinoacetate synthase, which is the same thing as this, and then we're going to talk about its regulation and some important implications. Okay, so basically I'm going to do the mechanistic steps in green like I normally do. Okay, so the first thing that's going to happen is a proton transfer and a nucleophilic attack. So this nitrogen of the guanidino group is going to attack the proton on a critical cysteine residue that's in the active site, and that's going to induce nucleophilic attack on this effective shift base carbon right here of the guanidino group. And what that's going to do is it's going to generate a tetrahedral intermediate as shown right here. Now remember that this is not a normal nucleophilic acyl substitution because this double bond is not between a carbon and an oxygen, it's between a carbon and a nitrogen, which makes that functional group that I'm going to circle in orange. This functional group is a protonated shift base. So it would be incorrect to call it a nucleophilic acyl substitution because it's not a carbonyl, but it's going to operate by a mechanism that's pretty much identical. Okay, so what's going to happen now is this shift base is going to reform. So this lone pair is going to kick in here to form the protonated shift base, and that's effectively going to expel ornithine. So this business over here, this business right here, this is basically the ornithine group, and usually they abbreviate it as L-ornithine, like this. Okay. L-ornithine is an amino acid. It's basically lysine, except for the fact that it is one carbon short of being lysine. Notice how you have effectively 1, 2, 3 carbons right here, and then the amine. If there was one extra carbon, it would basically be lysine, okay? But this is L-ornithine, and L-ornithine is basically going to go into the urea cycle, okay? As we'll find when we talk about the regulation of this enzyme, glycine amidinotransferase, we'll find that ornithine has one other function, okay? But for now, let's just say that ornithine eventually is going to end up in the urea cycle, and it will react with ornithine transcarbamylase, okay? Now, what this mechanistic step effectively did is it activated the amidino group. So this right here, this functional group right here, this is called the amidino group. And so the reason the name of the enzyme is glycine amidinotransferase is because what we're going to do is we're going to transfer this amidino group from the cysteine residue onto glycine. And so to do that, we're going to use a critical histidine residue that's in the active site. And the histidine effective Schiff base is going to do a proton transfer with the alpha amine of glycine, and that's going to induce nucleophilic attack on this Schiff base carbon of the amidino group. Okay, and that's going to generate, once again, a tetrahedral intermediate as shown right here. Now, the tetrahedral intermediate will then collapse back down to form the effective protonated Schiff base. Of course, there's a resonance contributor there, but this lone pair is going to collapse back down, reform the protonated Schiff base, and that's going to expel a leaving group, which in this case is going to be the cysteine thiolate, which will come over here and abstract a proton from this nitrogen, and that's going to effectively reform this functional group right here, which once again we're going to call a guanidino group. This is a guanidino group. And notice this guanidino group is attached covalently to this functional group over here. Well, what's that functional group? Well, that's called an acetate group. Okay? So the full name of this molecule that we form is called guanidinoacetate. And hopefully that name makes a little bit of sense. It's an acetate group, a 2-carbon carboxylate that's attached to the guanidino group. Okay? And what ends up happening now is we have this protonated imidazole, this protonated histidine, and what's going to happen is an inferred return step in which water is allowed into the active site and it reabstracts this proton from the histidine, and that regenerates the deprotonated histidine in the active site. Okay? But our main product that we're really concerned with is guanidinoacetate. And I'll go ahead and tell you this before we get into the regulation of this enzyme, that this guanidinoacetate is going to go react with guanidinoacetate and methyltransferase, and that reaction will give us creatine. Now, we've just seen the mechanism of glycine amidinotransferase, also referred to as guanidinoacetate synthase, but I want to talk a little bit about some problems that might be associated with deficiency of this enzyme, because it turns out that glycine amidinotransferase can be deficient in some people. And basically what ends up happening when you have deficiency of this enzyme is you can get things like neurological degeneration due to failure of energy metabolism. We'll talk about the energy metabolism based on creatine in the next video. But you can get failures of the brain due to neurological degeneration, which can lead to things like mental retardation. It can also lead to things that resemble autism. It can lead to language disorders. It can lead to epilepsy and things like that. So it's overall definitely not a good thing to have. Or in this case, you don't have it because you have a deficiency. Okay. Also, deficiencies and problems with this enzyme have been associated with end-stage heart failure. And what they found is that if you take the end product of this pathway, which is creatine, and you supplement that, it can actually lead to a positive outcome in the end-stage heart failure and at least alleviate some of the problems associated with that disease. Okay. And those are some of the sort of the maladies that are associated with deficiency of glycine amidinotransferase. Let's talk about the regulation. It turns out that there are basically 3 levels of regulation of this enzyme. The first is hormonal, okay? And there are several hormones that regulate this enzyme. Number 1, there's growth hormone. Okay, and I think growth hormone, when we talk about it, should make sense. What does growth hormone do? Well, growth hormone, first of all, increases bone density. So, like, for instance, it induces ossification. It makes you, gain height, which would be a direct result of bone size growth. It also increases muscle mass. And so if you think about it, if you have more muscle mass, you're going to have more demands of energy from those muscles. And so it would make sense for growth hormone to upregulate this enzyme, and in fact that's what it does. Okay, growth hormone upregulates glycine amidinotransferase so that you can make more creatine to supply the energy demands of the increased muscle mass. Okay, one thing that's important to realize about this enzyme is it's the committed and rate limiting step of the pathway. And so as we've seen in various pathways, usually the most regulated steps are the initial steps, usually the first two steps of the pathway. And this is no exception. This is the first enzyme of the pathway from arginine, and so it's the most heavily regulated. So growth hormone is one regulatory element for this enzyme. Also thyroxine, which is also referred to as T4 or tetraiodothyronine. That's a thyroid hormone, an iodine-containing compound, which basically works synergistically with epinephrine or adrenaline, and it increases the metabolism of cells. Okay, so if you have high metabolism by cells due to thyroxine, it would make sense to make more creatine to supplement that energy metabolism. Okay, because if you have high amounts of metabolism due to thyroxine, you need more of this energy-yielding molecule to create creatine phosphate to supply the demands of the cell. Okay, there are some other things that can upregulate this enzyme, like androgens, testosterone being the most notable one, because testosterone, of course, is the most potent androgen apart from dihydrotestosterone. Okay, but both of those hormones, DHT and testosterone, upregulate this enzyme, which leads to creatine synthesis. But one of the things that tends to downregulate it is estradiol, which is an estrogen. Now, the mechanisms by which these four hormones affect the levels of glycine amidinotransferase remains to be elucidated. We don't know if it is due to, stability changes in the mRNA that codes for glycine amidinotransferase, or if it's a direct increase in the synthesis of this enzyme. So that remains to be elucidated. There are also some other ways that this enzyme is regulated. Another way is through feedback inhibition. So it turns out that this product that came out in the second mechanistic step, this product right here, which is termed L-ornithine, okay, this guy right here, he can come back and he can feedback inhibit this enzyme. Okay, so ornithine can come back, apart from going to the urea cycle, it can come back and feedback inhibit glycine amidinotransferase. So in terms of arginine metabolism, whenever you're degrading arginine, we saw the degradation pathway for that in another playlist, where we basically did arginase, which gave us ornithine. Whenever you're doing lots of arginine degradation, whether it be to ornithine or to creatine, a high level of ornithine in the cell is a good indication that you have lots of creatine. High levels of ornithine are a good indication that you already made lots of creatine. So it would make sense that if you theoretically already have enough creatine, that ornithine could come back here and feedback inhibit this enzyme, and it turns out that that's in fact what happens. Okay? And basically the last level of regulation is through creatine itself. It turns out that creatine and some related molecules like cyclocreatine, which we won't talk about here, they can actually act as repressor molecules to repress the synthesis of the mRNA that codes for glycine amidinotransferase. So they're acting at the DNA level. They go basically into the nucleus, and they can basically prevent RNA polymerase from binding. So basically they're acting as repressor proteins to repress the synthesis of glycine amidinotransferase, and therefore you don't get the mRNA as much, and therefore you don't get the protein synthesis as much. So those are the three levels of regulation of this enzyme. You have hormonal, you have ornithine feedback inhibition, and then you also have creatine and cyclocreatine repression of the mRNA synthesis that codes for this enzyme. Okay, so basically at the end of this reaction, okay, we got guanidinoacetate. So now what we're going to do is we're going to come down here and we're going to look at the mechanism of guanidinoacetate N-methyltransferase. So this molecule right here, you would recognize this as guanidinoacetate. So this is guanidinoacetate. And essentially what we're going to do is this proton that's right here on the guanidino group, we're effectively going to replace that with the methyl group, which is part of this molecule, also referred to as S-adenosylmethionine. Okay, S-adenosylmethionine is the universal methyl group donor, methionine. Okay, S-adenosylmethionine, or SAM, as it's sometimes referred to as. Also, you can see the abbreviation as ADO-MET. Okay, this is the universal methyl group donor, and the methyl group that's going to get donated to guanidinoacetate is this one right here. Okay, so these type of reactions are basically bimolecular substitutions, or SN2s. So this reaction right here, this is going to be an SN2 reaction. Okay, so guanidinoacetate is going to get that methyl group. Let's look to see how that occurs. So in the active site, we have this critical aspartate residue. And I'll do the mechanistic steps in green once again. So aspartate is going to deprotonate the guanidino group there, and this bond right here is going to split. And it's going to, the electrons are going to come out here, and they're going to nucleophilically attack the methyl group of S-adenosylmethionine, causing loss of a leaving group. Now, what's important to realize about S-adenosylmethionine is when you attack that methyl group and you get loss of a leaving group, your leaving group is always this guy right here. This molecule is called S-adenosyl homocysteine. Okay. You'll also see this molecule abbreviated SAH. That stands for S-adenosyl homocysteine. Okay. Notice how right here the methyl group is gone. It got transferred onto guanidinoacetate. So this molecule right here, this guy, this is creatine. Okay, so this is a fairly unusual biosynthesis because it's only 2 steps. Very rarely will we ever see a synthesis that short, and so that's our final product. Our final product is creatine. Okay, and what we'll find is in the next video, when we look at the reaction of creatine kinase, is we can actually transfer the gamma phosphate of ATP onto this molecule to create something called creatine phosphate. And just to finish up the mechanism, we have this now aspartic acid in the active site, and we need to regenerate the resting state of the enzyme. So just like in the case of glycine amidinotransferase, we have an inferred return step in which water comes in and re-abstracts the proton from the aspartic acid, generating the fully deprotonated aspartate residue in the active site. Okay? And of course, these get released, and creatine can now serve in energy metabolism, and we'll look at the metabolism of that in the next video. Okay, and just to remind you that this creatine still retains the guanidino group. The guanidino group is this guy right here, except the guanidino group now has a methyl group attached to it. And of course, we still, I'll do this in gray, we still have the acetate here, okay? Another name for this molecule would be N-methyl guanidinoacetate. N-methyl guanidinoacetate. But usually the common name that people give is, of course, creatine. Also, we can talk about defects in this enzyme. They're not as common, but they can lead to some of the same neurological symptoms that you would get if you have a deficiency of glycine amidinotransferase. So you can get neurological symptoms that can produce things like mental retardation, symptoms of autism, and so forth. So there aren't any good things that come out of a deficiency of any of these 2 enzymes. But like we saw in the first case of glycine amidinotransferase, one of the ways that you can sort of fight against a deficiency of these guys is to supplement creatine. Now, one thing I want to talk about, and I'll mention this in the next video as well, is that there are a lot of supplements on the market that you can sort of buy in drugstores, and many of them are really not purported to work very well. There's limited evidence on them. Sometimes there's no evidence at all. They're just sort of kind of superstitions that people have, and they take these supplements and they think they work, and maybe it's a placebo effect. Sometimes they don't work at all. But creatine is one of the few cases out there for supplements that is known to work. Because creatine is a natural biomolecule that's produced in humans, you supplement this and then you eat a large meal. Of course, when you eat the meal, you end up producing a lot of adenosine triphosphate. And then, as we'll find, you take the creatine, and through a reversible reaction catalyzed by creatine kinase, when you load up your cells with ATP, and of course they have creatine right here, the gamma phosphate of ATP will be transferred onto creatine, yielding ADP and creatine phosphate. And the creatine phosphate basically stores the high energy gamma phosphate on it, so that ATP is not free to allosterically inhibit a lot of the enzymes. Plus, if you generate ADP, you can use ATP synthase to make more ATP. So the creatine basically serves as a reserve for that high energy phosphate, and then once you need the energy, you basically go back this direction and reproduce the ATP, and of course this guy is burned by muscle cells and by neurons using their sodium-potassium ATPases. Okay? But like I said, this is a supplement that is known to work. And just remember that if you are intaking a lot of creatine, you don't need to be making more. So what will end up happening is creatine will come back into the nucleus at the DNA level, and it will come over here, and it will basically repress the synthesis of the mRNA that codes for glycine amidinotransferase, therefore getting rid of the enzyme that catalyzes the committed and rate-limiting step in creatine synthesis. Okay, so I hope this video gave you a little bit of intuition on the synthesis of creatine from L-arginine. In the next video, we'll look at the physiology of creatine more closely, and also the mechanism and physiology of creatine kinase. See you in the next video.